Hi everyone and welcome back. So now we're going to talk about different types of infections associated with animal viruses. So we can have acute infections, we can have persistent or chronic infections, and then we can have infections that are called latent. An acute infection is one that has a very rapid onset. Typically it's very short in duration, but it's really intense versus a persistent infection, which can be continuous for years and years, and can show symptoms at times, and then other times may be asymptomatic. So here's an example of an acute infection. Influenza has a very short incubation period, anywhere from 24 hours to about 72 hours, and then has a very short span before the virus is eliminated from the body. This can last from 7 to 14 days at the most, typically, and then the virus is gone. But during that period, the disease is very debilitating on the individual. And so we're not talking about a stomach flu, we're talking about respiratory influenza versus a chronic infection or a latent infection. So now I'm going to get into chronic and persistent are very similar. So again, I talked about a persistent infection being one which you might have symptoms at times, and at other times you may be asymptomatic, but it's continuous. So a chronic infection is one in which you always are releasing low levels of viral particles. So you are able to transmit the infection continuously. A latent infection is where you have the infection in the cell, but it's like a provirus. So it's sitting there and it's not causing any problems. And then every once in a while it reactivates. So here we see hepatitis B as a chronic infection. So it's always contagious, but it doesn't always cause problems for the individual, but the individual can pass on the disease. A latent infection is something like cold sores, the herpes virus. So cold sores come and go, and when they come, they're typically acute infections, so they last for maybe a week or 2 weeks. But then they go away, and during the period where they are not around, where you don't have any infection, you're not infectious either, so you can't spread the disease. And here we have a reactivation of the virus. And typically reactivation of the virus can occur because of stress or other problems. And so you have these outbreaks of the disease that then go away after a week or so, and then they come back later in life. That's latent. And so here you can see some different disorders that are either latent or chronic. And then HIV has both chronic as well as latent infection. The last thing we're going to talk about with viruses is how we grow viruses. So viruses have to have living cells to grow. They can't grow on their own because they don't have any metabolic properties. So infected cells are grown typically in solutions with the virus in the solution. And then filters are able to remove the virus from the cells. And so here we have a filter. This is a figure from my OpenStax textbook. And here you can see animal cells, you can see bacterial cells, and you can see viral particles. And so we're releasing animal cells and viral particles. We're trapping the bacterial cells. Or I'm sorry, I was like, wait a second, that doesn't make sense. We're releasing the bacterial cells and the viral particles, but we're keeping the animal cells up here. And then we can filter this new stuff, and we can filter just the virus out. So depending on the size of the pore would filter. Viruses are much smaller than bacteria. So we can use different size filters to filter what we want. So in vivo studies, we use animal embryos to culture viruses. Examples, we use chicken eggs to help us in producing different vaccines. And we can also use turkey eggs. In vitro studies use animal cells, so not animal embryos, but animal cells. One important cell line is known as the HeLa cell line. HeLa cells come from a woman, here you can see her, this is Henrietta Lacks. Her cells were taken by her doctor. She had cervical cancer and she died of cervical cancer. Before she died, her doctor did remove some cells from her cervix, and then he sent them to a lab, and that lab has kept her cells alive. They're still used today. So this was back in the 1940s. Her cells are still used today because they are known as the immortal cell line. In studies on polio, on cancer, including cervical cancer, which my daughter actually studies, the HPV virus and cervical cancer. They've also been used in studies for HIV and AIDS. Then we have plant viruses. Plant viruses are economically important because they damage plants, which we need. So they can damage food, cause food shortages, and they can cause problems with other animals so maybe not food shortages for us, but food shortages for other animals, as well as pollinators. Because they are typically transmitted by insects or through the soil, as well as through pollen. It's hard to eliminate these diseases. An example would be like Dutch elm disease is a plant viral disease. Plants also have to worry about viroids. So viroids are short strands of circular RNA that are able to replicate on their own. These were discovered by Theodore Diner in 1971. And these are obligate intracellular pathogens that only infect plants. And over here you can see potato spindle tuber viroid, and the little tubers that these potatoes have because of the disease. Lastly, we have prions. So prions are proteinaceous infectious particles. They were identified by Stanley Prusner in 1982. He was studying Scrappy in sheep, and he identified these, and he eventually won a Nobel Prize for his discovery of prions. So there are a lot of different diseases associated with prions. So this is a list of different diseases associated with prions. Most people probably have heard of Kuru. So this is the cannibalistic disease, bovine spongiform encephalopathy, and Creutzfeldt-Jakob. Those are the ones that most people talk about a lot. So a prion is basically an infectious protein. It's a protein that is misfolded, and it can cause other proteins to misfold as well. And so this is a brain that doesn't have any prions, and this is brain tissue or nervous tissue with prions. You can see these large spongiform lesions that are found that are going to cause all kinds of problems. There's no treatment for prions or for a prion disease. You just treat the symptoms. An individual that has a prion disease is going to die. That's a kind of sad ending, isn't it? But that's the end of my lecture. So be thankful that you have a healthy brain, and I will see you in our next videos. Have a great day. Bye.